In this video, we will discuss about this pancreatic cancer progression and the glycosylation perturbation. We have discussed many times how these pancreatic cancers progress in several videos. Today we will discuss this pancreatic cancer cell progression and how or at which or how this glycosylation perturbation also begins, because we have seen that in the pancreatic cancers there is a lot of truncated O-GlcNAcs which are not found in the normal pancreatic cells. So we will start with this mutation landscape. So what will happen? This pancreatic cancer progression usually begins from this acinar cell, and there is what happens in the acinar cells. There is DNA damage occurs because of some of the mutations, and this DNA damage mutations leads to the transformation of this acinar cell. And at this point of time, what will happen? The mutations leads to the uncontrolled growth rate, and it will also cause genome instability. And till this stage, through this immune surveillance or EDAC, that is epithelial defense against cancer, these mechanisms of the immune cells, it can remove these genomic unstable pancreatic cells. If cells are able to survive or pass through these stages, then it will lead to the sustained proliferation and stromal dysplasia. This is how the pancreatic cancer progress. So what are the mutations that usually occurs or that people have found out generally in the pancreatic cancers is this KRAS mutations, and they, as the pancreatic cancer progresses, it will leads to these CDKN2A mutations and TP53 and SMAD4 mutations. And these TP53 mutations prevents the cell death, as when the cells have like uncontrolled behavioral changes, then TP53 will decide that the cells now will go to the program cell death, or there is a possibility, if there is a possibility that the cancer cells or cells can be moved back to the normal state, then it can prevent the cell death. This is the job of TP53 mutations. But if it is mutated, then it will never go towards the cell death, and this uncontrolled growth rate continues to occur. So this is one of the like special functions of TP53. And how what are the different stages that are defined based on this pancreatic cancer progressions is that first, this acinar to ductal metaplasia formations. After that, this panc interstage panc 1N stage formations, or basically it is the intermediate stage, and then it will go to panc IN2, panc IN3, and thereafter this pancreatic ductal adenocarcinoma. And at this stage, the pancreatic cancer acquires this high migration properties, and it has begun to spread across, or it has already spread across different parts of the organs. And from the glycosylation perturbation point of view, what will happen is that as they move towards this uncontrolled growth rate, or the metabolism rate increases, there will be a switch in the pathways that it needs high glucose consumptions. So it will switch to high glucose aerobic pathways, and that is also known as this Warburg pathway, popularly known as Warburg pathway. And as they move towards this glucose perturbations, it will also cause this hexosamine biosynthesis pathways upregulation, and this will lead to the O-GlcNAcylation of this enzyme PFK1. And this O-GlcNAcylation of enzyme PFK1 will reduce its efficiencies, and this will act as a positive loop or positive control, and it will further accelerate these HBV pathways to promote O-GlcNAcylation. So overall effect will cause the high UDP-GlcNAc and high O-GlcNAcylation within these pancreatic cancer cells. And this high GlcNAcylation and high O-GlcNAcylation thereby promote this increased TN antigen, and this increased tumor silations, and the increased glycoimmuno checkpoint. One of the glycoimmuno checkpoint that we have discussed in one of our videos, one of our paper that we have discussed, is about the Glycan interactions with the pancreatic cancer selectants on the immune cells, interactions with the ligands on the pancreatic cancer cells, and that ligands are related to this integrin subunit. So if you want to refer to that video, you can go to that video, or if you want to understand more about this pancreatic cancer perturbations and glycosylation perturbations in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.